Systems biology is about identifying the pathways that underlie a complex trait. The trait may be metabolism, how an organism tells time in the biological clock, or cancer. There are three challenges. The first is that the models that describe how pathways function involve many parameters, and we have limited data. We introduced Ensemble methods to overcome this problem, and these methods come from statistical physics. The second problem is one of scale. Usually pathways describing complex traits involve hundreds, if not thousands, of genes, and so any procedure that you have for identifying those pathways has to scale to involve thousands of genes. And the third challenge is that we often don't know the shape of the pathways, who regulates who directs who. And that's the problem that we have solved here and we're focused on today. The example that will illustrate the solution to this problem is for the biological clock. The biological clock is best understood in a model fungal system called Neurospora crassa. And here's an example of the biological clock. The complex trait of interest here is the organism growing from one end of the tube to the other end of the tube. And every 24 hours, these white bands appear. That's the organism reproducing. The white bands are the spores that give rise to new individuals in the tube as the organism grows down the tube. And that banding pattern is the biological clock. The challenge for us is to identify the thousands of genes that are involved in controlling this trait.
The method that we're using to accomplish this model identification is borrowed from statistical physics. We refer to it as the Ensemble method. And the idea is that we want to actually not identify one unique model, but a whole statistical ensemble, a large statistical sample of probabilistically determined model parameterizations. Unfortunately, this method does require a repeated solution many times over of the underlying differential chemical rate equations. And with that, we are forced to go to a very large-scale massive parallelization in order to handle very large biological systems of the sort that we're interested in.
This is the CUDA Research Center in the Computer Science Department at the University of Georgia. This center has 30 GPU cards that have been donated by NVIDIA. Using the structure of the genetic network, we are able to subdivide the big problem into lots of smaller sub-problems that can be solved efficiently in parallel using GPUs. This approach has shown that the Ensemble methods are scalable.
The key to identifying the network of genes when their connectivity is not known is the supernet, where all of the genes in boxes are assigned equally to the regulator in circle. And as the simulation runs and proceeds, we get the infrared net where all of the genes now are really assigned to their putative regulator. Here's an example of the gene assignment where each dot represents a gene and each color represents a regulator assigned to that gene.